All this is Dr. Mobeen Sayed. Welcome to one more show. So I was having post Thanksgiving fun time. I hope you had great Thanksgiving as well. And then this concern about the Omicron or this new variant started surfacing everywhere. So I thought this is important time. I saw some of the messages, I saw some of the videos, and the fear mongering just started. And I thought I should try to understand by for myself as well for how scared we should be, and then discuss it with us together over here as well. So the way to approach today's discussion is to look at the data together. Then what is your opinion? Is your opinion? I'll give you my opinion as well. And these are, of course, opinions at this time. We'll see how it unfolds. But uh, I think that the things are not as bad as they look. So let's start. Not as bad as they look, or not as bad as some folks are uh, making it appear. And I want to show you a specific chart to clarify how some folks are using a chart to create even more nervousness. So here is drbeen.com. Black Friday sale is going on. This is the World Health Organization, and they have called it variant of concern, and there is a reason for why they called it, and we'll talk about that. This is a very good article in New York Times, and I saw a friend who also does these talks on the internet, and he had just verbatim gone over this article and narrated it. So it is a good article. Then this is very interesting. This is B11529, the variant we're talking about, and the 30 mutations on the spike protein and more than 50 or 50-ish mutations overall. And what is their role? What could be the concern there? So this is a good article as well. All the references are in the description. Then this is the important thing that I wanted to discuss. So let's put this in front of us. So if you wanted to just look at this and say, okay, fine, I don't want to hear the rest of the talk, then that is totally up to you as well. So look at this. This is South Africa. Again, in my opinion, we cannot really say it is South Africa. It may be Botswana. We can't even say if it is for sure just the Botswana. So regardless of where it came from and how it traveled where, South Africa has been nice enough that they sequenced it, they informed us, and here we are. So check this out. Uh, in this chart, down here is so look at the chart. It is daily new confirmed COVID-19 cases. Good. Down here we are saying these are the previous three peaks. So for example, in July 20, 2020, 12,261 daily cases. Then here in January 10, 2021, 18,692.71 daily cases. And then, or 7 days rolling average, not daily, 7 days rolling average. July 8, 2021, 19,955, 7 days rolling average. So these are three peaks. And now South Africa was at about 264 daily 7 days moving average daily cases, or 7 days moving average cases. And now they are today, for example, are at 1,000. Yesterday they were at 600, and before that about 300, and so on. So that is the change from 200 to 1,000. So if you put that as a percentage change, then this particular change looks very similar to the previous changes because it is percentage. For example, if I said that a number changes from 5,000 to 10,000, then we say it is doubled. And here it is showing as doubled. And if I say a number changes from 2 to 4, that also is a double. So if you just put the percentages or rates in front of us, then it would look like that this current peak is very similar to the peak before, or even has already topped it. But actually, the actual rates are low. We will continue to see, of course, in the coming days. Nobody has seen the future. We'll continue to see the coming days that how does this change. That is number one important reference to keep in mind. So whenever you see this chart, please be more careful and go back and look at this chart as well, maybe on daily basis for the coming days. I'll be off tomorrow, day after. Just look at this chart to the absolute numbers too. This is next strain, and of course we know that there are thousands and thousands of SARS-CoV-2 strains or variants. This is not the very first variant. This is a variant of concern, and I'll explain why, but there are all the variants over here. And these are, I always read this part down here where they say there are thousands and thousands of them. We can only show 3,000 for the processing powers problem, and that is all we are showing at this time. So huge number of variants are already there. This is GISAID, the unique mix of spike amino acid changes in Omicron is of interest as it comprises several that were previously known to affect receptor binding and antibody escape. So meaning it does have changes that are very similar to previous variants changes too. Now please remember, these large number of changes are also a possible detriment for the virus too. It has to stay stable with all these changes. It has to stay viable. It has to stay infectious. And if it wants to be lethal, it could. Although I would propose virus is going to continue to learn how to live with us. So it is going to keep reducing the lethality, or the selection pressure on the virus will make sure that that virus variant survives, the one that causes less damage, because only then it can continue to go from one person to the other without causing too much of a damage and still survive. Otherwise, it would die with the person. This is an important page, and I'm going to come back to this page a little later. I want to look at these changes in the virus protein with you. Over here, this, just for the time being, this green one here is the ACE2 receptor, and this other big blob is the spike protein. All these orange changes are the changes of concern. Green changes don't really matter too much. Cyan changes may be also of concern. So I'm going to look at these changes with you in a few minutes. This is another quick article, not quick article, it's a big article, but anyways, they talk about that S1 unit and the receptor binding unit can become on to bind with the RBD, with the ACE2, or it can become off to not bind with that. So it can be an up or down state. And then what does Omicron mean? Omicron, it is actually O little. Omega is O big. So these are the Greek letters. Omicron is O little. Micron means small, or really very, very small, and mega means big. So I believe that there is Omicron, Omega, and OU as well. So this is Omicron. It is also, I believe, the 16th letter, 15th letter of the Greek alphabet. And this is an interesting article as well, if over the weekend you wanted to read it. N-terminal domain mutations of the spike protein are structurally implicated in epitope recognition in emerging SARS-CoV-2 strains. So we'll talk about these a little more. Now, with these references that are present, and the links are in the description too, let's start our quick discussion. So Omicron, literally the little O, it should be 15th letter over here. I called it 16th or 15th letter. That is done. So B11529, it is said that it was first seen in Botswana. And in Botswana, there was a lab where there was an HIV patient's blood sample in which this was seen. And when they were sequencing it, they found that this is strange, that this has 50 changes in it or mutations in it. And that is how they further made it aware. Then South Africa showed it to the world as well, or reported to WHO and so on. So possibly from an HIV patient. I am already seeing articles and comments and tweets and things to say this is because of vaccination and this is not because of vaccination, or somebody who's not vaccinated did it. Look, the variants can occur. We don't even know how prevalent this variant will become and how lethal will this be. So I think it is premature at this time to start looking at those. The discussion today, which we are going to do, is we are going to look at the variant itself and kind of start forming an opinion that how nervous should we be. I think we should not be too much nervous, but let's look at the data. Your takeaway may be different. You might come back and say, no, I want to be more nervous. I think there is more concern, and that is fine. So, first reported to WHO on 24th November, was reported in a specimen collected on 9th November. And about 50 or more mutation, we called it little o. So they are these guys sitting over here and saying, you called us little o, we'll show you. So that's just me. Okay, so designated variant of concern. Why? From the WHO's point of view, those things here, if we look at this last part, this is their definition of variance of interest and variance of concern. So variant of concern is a variant that might have any one of these. Number one, it increases in transmission. Then they'll call it a variant of concern. Or it has detrimental changes in epidemiology. For example, we saw in South Africa doubled the transmission, or 4 times or 3 times, and all of that is a, if you look at the rate, then you would say, well, that is a significant change in epidemiological behavior of the virus. What will be very interesting is to see those 1,000 patients, for example, today, what is their clinical progression. I would suspect that if this virus has the same uh, changes as Delta plus more, and it stays stable, then it will actually become a replacement for Delta. The question is, will it be more lethal? If it is more contagious, if it binds faster and enters the cells faster than Delta, then that window of time for incubation period might reduce. If that reduces, then it would have a problem. It would create a problem because it would just be very rapid in causing symptoms, and our immune system may not be able to catch up that fast. However, if the incubation time becomes longer, then it would be a dud. It would not be able to do much, because if it is sitting out there for 10 days, very slowly increasing in replication and increasing viral load, and after 10 days it causes enough damage that symptoms appear, then it would really not become too big. Or you could say it might become too big in terms of presence in people, but might not be as lethal, which is a good news. Okay, so detrimental changes in epidemiology, we are seeing that in virulence. So increase in virulence or change in clinical presentation, that we don't know yet, right? So it's just being reported. Reduction in effectiveness of public health and social measures, for example, social distancing, masks, and those, or hygiene, or available diagnostic and efficacy products and their change. So again, on the diagnostic side, there is a change as well. But please remember this. This is what I was saying, that folks would continue to pick up various sentences and rumors will be created. This is why I actually came in today to have this discussion. So look at this. This is WHO, and they're saying current SARS-CoV-2 PCR diagnostics continue to detect this variant. Just this one statement should speak volumes over every other concern. And the reason for that is, if this variant has escaped, then the tests would fail very quickly. So that is one. Then they say, but there is one test. So several labs have indicated that for one widely used PCR test, one of the 3 target genes is not detected, which is called S gene dropout or S gene target failure. The point is, every test has 3 signal areas or 3 type of antibodies that would bind to various parts of the virus. So if I go down here for a second, there may be antibodies in the test that would bind to various parts of the virus. And I have only made spike proteins over here. There will be an antibody that might bind to N protein or M protein. Every test has a different structure. But they try to bring in antibodies or nuclear genetic gene detection signals that are unique to this variant. Otherwise, they'll start picking up human coronavirus from our nose and throat and say you're positive. So they have to be very, very precise for this virus. And what we are seeing is that Majority of the tests are working fine. That means these instabilities, these mutations, these changes are not big enough to, to fail the tests. And if they're not big enough to fail the tests, they're not going to be big enough to fail the efficacy of a previously infected person's protection, or a vaccinated person's protection, or monoclonal antibodies protection, or other drugs protection. So this is not a SARS-CoV-3. From this one sentence, I could extrapolate that. Maybe I'm wrong. We'll see more. But I don't think this is SARS-CoV-3. I don't think this is going to be now the it could be replacing Delta, but I don't think it is going to start a new pandemic. Okay, so let's continue to read here. So they're saying, and this test can therefore be used as marker for this variant pending sequencing confirmation. Remember, this happened in UK as well, that in UK, when the Delta variant appeared, one of the tests failed. And on that test, two signals would continue to work and one signal would fail. This is exactly the situation here as well. All tests are working, one test is failing, and even on that one test, two signals are working and one signal is failing. That one signal is on the spike protein. So that those mutations are on the spike protein as well. And UK had used that kind of a thing to identify deltas. Similarly, WHO is saying you could use this test to identify Omicron. But there is a ray of light in here. All of the tests are working. That means it is not enough change there to fail the tests. And if the tests are not failing, then other things would also not fail. So continuing with our discussion. Going back here. So change in virulence and clinical features that we do not know yet. Effectiveness of the public or social measures, we will find that out. Diagnostic change, there is at least one test that has become changed. So because of these things, that there is a epidemiological change, that there is an increase in cases, rate of cases has increased. Absolute numbers are not very high, but rate is increased, and the of course it is spreading to the other places too. Now please remember, all variants do spread to other places as well. It has gone to Israel as well, it has gone to UK, it has gone to Egypt and Hong Kong, and or from Hong Kong or from Egypt. So it has been circulating now. US put some restrictions in, UK has put restrictions in, other countries are putting restrictions in. I think that we should still continue to make sure. If I am the one who is looking at it, I would ask, the very first thing I would ask, let's say WHO, I'm sitting somewhere in WHO and I want to make sure that these things are, I'm aware of what is happening. I would ask them to find out the clinical outcomes. Find out how fast or slow the patients are deteriorating or not deteriorating. That would tell me immediately at least an idea of what this would do. From that one statement, I can already extrapolate that this is not going to be a hugely damaging virus variant. Anyways, we'll see. Now, continuing. Concerns. What are the concerns with this variant? And I'm going to go to the changes in a second. I want to first put things out in front of us. So 30 changes on spike protein. 30 changes on spike protein. That you can say really bad. It's going to be really tough on us, right? It's going to bind fast, and I'll explain why. But at the same time, please keep in mind, these kind of changes can make viruses unstable as well. These changes can make the viruses incompetent as well. So maybe it is able to bind faster, but is it continuing to have its lethality? Because you know that there is a lot of lethality of the virus itself imparted by the spike proteins pathogenesis, or spike proteins capability to cause pathology. What if these changes, fine, it allows the spike protein, to tightly grip the ACE2, and because of that there may be cellular inflammatory imbalances, so that is a pathology. Or tightly grip CD147, we don't know, but that would be a pathology. And then spike has more ways to cause pathology. Maybe those ways would not work that well. Maybe this will become a more humble virus in terms of disease, but more efficient virus in terms of binding with the receptors and entering the cells, that would actually make it a better virus for us. So we'll see 30 spike protein mutations. There is an important mutation that is P681H. This is a mutation at the boundary or the furin cleavage site. What does that mean? We know that when this virus arrives, SARS-CoV-2, when it arrives in our body, the spike protein binds with the ACE2. Then the TMPRSS2 sitting on the side cleaves or primes the spike protein by cutting it. So where it cuts it, that is a furin cleavage site. There is a change on Delta variant, that is P681R change, that made it easier to be cut, which allowed it to become more efficient in work because it would be just easy to break the spike protein and prime it. A similar change is here. So it is possible that this virus would also become easier to prime. Fine, Delta was easier to prime as well. Then there are some more changes. Not all changes are interesting, but there are some more changes that were also present in the previous variants that are more efficient. That means it is really just equal to their efficiency so far. We'll see more. And that means it is possible that its binding efficiency. Binding efficiency is the magnetic forces, and magnetic, these are really chemical or electromagnetic forces, positive and negative ions present on the protein of the spike protein and the ACE2 and their binding affinity to each other, plus steric affinity, that is shape affinity. That is, if my hand is like this and the other hand is like this, and this is how they fit together, this is the shape. But if this guy has a different shape, then they cannot fit together. Or if this has a shape like this, they cannot fit together correctly. So maybe the shape fit may become better. And not only shape is important, but electromagnetic forces are also important. You may have a shape that fits very well, but their electromagnetic forces, if these are repellent, then it would not work. So electromagnetic forces plus shape forces, they both have to work together correctly for a better binding. So we'll see how it goes. So binding affinity may increase, and then transmissibility may increase. Transmissibility, so binding is entries. You bind to ACE2 and you enter the cell, this SARS-CoV-2. Transmission is that you come out of the cell and then you are cleaved off. The furin cleavage site splits the virus away from the cell, and the virus then goes and do other things. Remember, furin is not just for the spike protein priming, it is also for the cleavage of the virus away from the cell. So it may actually become a virus that would enter cells fast, that would exit cells fast, and infect cells quicker. Now, if it started causing damage to the cells, and it is doing it rapidly, that in itself could cause more severe clinical outcomes, especially in those that are at risk. It could also cause severe outcomes in those who were previously infected and recovered, or had been vaccinated and have some time gone and their vaccine status has reached a point where their immune system is at rest. And now let's say this virus, let's assume this virus can create damage within 1 day instead of 2 or 3 days like Delta. Then that means our immune system would have to ramp up within a day to respond to it if we are previously infected or if we are vaccinated. And it's, in most people, it's not going to ramp up. Our immune system is not going to ramp up within a day. So how would then we handle this situation? The handling will be, in at-risk population, we might ask them to take a booster, and that booster might cause their immune system to be ramped up. In majority of the folks, at least from that one sentence, I'm hanging my hat on that one sentence, that most of the tests are not failed. I think in majority of the cases, the protection would continue as is continuing for Delta. Efficiency might be slightly reduced. Okay, so what would happen to infectivity? We'll see. I think it is increased. What would happen to load? Of course, if the entry is increased and exit is increased, and load would increase, and that should mean shorter incubation time. If we are saying that the incubation time is long, which I'm hearing from some folks that 8 days, 10 days, if that is the case, then its load is not increasing fast enough. And then it is going to be a dud virus, which is a good thing because it's not killing us. It is sitting in us, allowing the immune system enough time to respond to it and take care of it. So I'm not saying it dud as an insult. I'm saying it's a good thing for us. So I don't want it to hear and become angry. And then how about shedding? So again, it depends how fast it is entering and how fast it is coming out. So these are the changes on the spike protein. I want to continue because I want to show the spike protein changes themselves. So here, another problem. This problem is the previously infected or vaccinated or monoclonal antibodies or tests. So imagine this is a spike protein. On the spike protein, these are the antibodies that have been produced against that, either because of infection or vaccine, or these are monoclonal antibodies given. And they go and they attach to various parts of the spike protein, and they kind of wrap it, and they prevent it from binding with ACE2 or CD147 or other places, and kind of take care of it. Now, when we have these wholesale changes in it, when we have these wholesale changes in this spike protein on this side, then what happens is that these shapes the phenotype. Whenever you read, you're going to read about this virus a lot now. So the phenotype means shape, and the genotype means genetic material. So the shape may change, and that changed shape may prevent an antibody to connect with it. So here is another antibody, here is another antibody. They're trying to connect to it and they cannot because its shape has changed. But remember, if the shape has changed sufficiently, then the virus itself is going to have a problem doing its pathology as well. So it would have to preserve some changes and make some changes, and we'll have to have a balance out there. So far what we know from that one, from one test, that one test is failing on one gene, I don't think that there is too much of the change. So one antibody is failing so far. Binding site may change. Now, what else? So this is what is spike S gene dropout. We talked about it. Could drugs fail? So for example, drugs, IVM, or this drug from Pfizer that is coming in, they are binding, Pfizer's drug is RDRP. So any drug that may bind to the spike protein might fail if the changes are big enough to cause the shape change enough that the antibodies cannot work. I will continue to add this balancing statement that if the shape is so much changed, then it is possible that the virus itself would not work too much. And if the virus is working, then shape may not have changed that much. But anyways, there is a possibility that drugs could fail. Could monoclonal antibodies fail? Yes. Just like vaccine could fail, just like previously infected person's immunity could fail. Will they fail? So far from that one place where I've hung my hat, I don't think these are going to fail. So then what is interesting in this discussion? Number one, absolute increase in number is not that big. It is increasing. We'll have to see in the coming days to see, does it really become another huge spike? Remember, South Africa is about 1/4 or 28% vaccinated or one dose vaccinated. So it's not very vaccinated country. There are other countries that are very vaccinated. There are countries that are taking ivermectin. So we'll see what is the dynamics of this virus. If it becomes less lethal, less damaging, and becomes more prevalent, then I would welcome it. If this becomes more prevalent and more damaging, then we will have a problem. Incubation period seems longer. If it is longer, then it is actually less damaging, because short incubation period is actually an indicator of virus's damage. It is replicating fast and it is damaging the cells fast, and that is how we are getting symptoms very fast. The symptom of the virus or the disease is a representation of the damage that is occurring. So if a virus can create symptoms in a day, that means it is causing a lot of damage. But if a virus that creates symptoms in 10 days, that means it is very slowly causing damage. This virus itself could perish because of the instability, because of all the changes on it. And most tests are still working. And I want to show you this before somebody says that, hey, Mubin said it can perish because of instability. Look at this. This is a World from Molecular Forum. And here you might actually, variant of concern, could B11529 variant be unstable? The large number of mutations may make the virus unstable. And I've been saying this for the last whole year, that there could be three outcomes to the virus. The daughter could become more efficient, it could become the same way as the parent, or it could become less efficient. And the efficiency is not an absolute thing, that if it can bind better, then it is more efficient. It may be less lethal, but more efficient in binding, which is useless, but good for us. Okay, so this is the discussion done. Now what I want to do is I want to look at this spike protein itself. So please keep these two charts in your mind in the next coming days. Look at this chart as well. This is the absolute number. And absolute number is also, look, it was 290, 286, 264, and then it went to 500, so doubled. Then it went to 1000, doubled again. So there is a jump in that, but that is still a tinier thing. We'll see. When it did this here as well, it went the same way. So I'm not saying this will not happen. What I'm saying is keep an eye on this as well while you are looking at this rate change, because rate change already looks like a change like the previous spikes. But look at the absolute numbers too. We'll see how it goes. That is one. And then here is what I want to do. This is the this is the most important thing I wanted to do. So this is the spike protein. And this is the spike of this Omicron virus. This green thing here is the receptor binding domain. This is our cells RBD, ACE2. And down here, these orange changes are on the receptor binding domain that is interacting with the ACE2. So all changes here are changing the dynamics of the binding. They may make it more efficient. They may make electromagnetic forces to be better. They may make them to be bad. They may make the phenotype, the shape, to become better and a better fit, or they may make it unstable. They also may make, look at this, there are so many other, all these orange changes are the changes that may be of concern. So for example, there could be a change here on the orange side where an antibody was going to attach, and maybe that antibody would fail. Again, keep going back to that statement from WHO that most tests are working fine, and one test is failed on one gene, which is on the spike protein. So that means it's not that these changes are actually already wiped out the whole system's capability, our system's capability, our diagnostic tools capability. That means our protection is also not wiped out. That means vaccine or previous infection is not wiped out. That means monoclonal antibodies are not wiped out. That means drugs are not wiped out. But still, this is a plethora of changes. Look at that. And this could go bad for the virus as well, that it would just become unstable. So I think I'm repeating now because I'm just so excited to see this little new virus, and how I saw so many folks are just becoming nervous and making people nervous as well. So this is where we are at. I hope it makes sense. Your takeaway may be different. Maybe I'm missing something. Maybe things would evolve in the coming days and we'll see more. But I am not worried about it. Me personally, I'm not worried about it. My wife came to me today and said, "Oh man, what has happened? Do we have a pandemic once more?" And I sat down with her and looked at some of these things, and I said, "I don't think we are in that place yet." So I understand that there is a lot of concern, but at the same time, I think we are not at that place yet. And I wanted to give a shout out. So many folks in the Texas Meg has helped. John Snyder has helped. So, John, yes, for the for the mutations and for various links. So thank you very much. Now, if you are okay, I will skip the chit chat and go back to my vacation. But if you like, please comment here. I'll leave the comments on uh, after going offline, and I can read there if you would like me to come back for the chit chat. So thank you very much. Have a good day. Happy belated Thanksgiving. And if you would like to support this work, the easiest thing is to just like this video, and you can subscribe and share as well. And if you would like to do more than that, then there are 3 links in the description. You can buy me a coffee, or you can become a patron, or you can use PayPal. Thank you very much. Continue to be safe and happy and relaxed. And comment here if you would like me to come back. Bye bye.